Welcome back, children. Further, we are going to study the need of cell division. Cell division means one cell dividing into two new cells. Now, why the cell division takes place? There are three reasons. First of all, help in growth of the body, repair wounded tissues, replace old and worn out cells. This time of your age, you are growing. So how is this growth taking place in your body? That is with the help of cell division. If you get any wound, within few days it is repaired. Is because of the new cells which are coming up, and it replaces the old and the worn out cells. That is why cell division is necessary event in the life of all living beings. So you can mark these 3 points which are very important, that why do we need cell division. Now, how to study cells? You can underline cells are always studied with the help of microscope. To differentiate, identify various type of cells, we color them with some dye. So you can write here, with some dye we color them, and this dye is all called stain, and the process of dyeing it is known as staining. And the stained cells are mounted on a clean slide and observed under the microscope. Now this is your activity 1. This is to study plant cell in onion peel. Cut a piece of onion lengthwise into pieces and remove a fleshy leaf. Fleshy leaf is the part of onion which we eat. Actually it is a fleshy leaf stored with food. So you break it and peel off. There will be a very, if you try it at home, peel off something which is on that leaf. So you'll find a very thin membrane. So that thin membrane is taken from the fleshy leaf, and then it is kept on a clean slide, and then now we are going to use a dye. So that is, what is the dye here? A drop of methylene blue, or we have another, that is saffron 2, any one you can take. Now spread the peel with the help of brush and needle, and then cover it with a cover slip, and teacher will focus the prepared under the microscope. So this is what you see under the microscope. Can you see this? Rectangular cells. There's a clear space inside, and one cell is jointed with the other cell, and this dotted area, if you see this dot area, this is all cytoplasm. Okay, this is nucleus, and inside this is vacuole. So what is the observation? Brick shape. See, this is the structure of brick lying side by side. Each cell has unclear cell wall, a centrally placed vacuole, and a thin layer of cytoplasm between the cells and the vacuole. So draw a diagram and label the parts. So this diagram you can draw, and you have to label the parts. So the D part, right? Further, we are going to read activity 2. To study animal cell from human cheek. Now this is again, what you are going to do is rinse your mouth, that is with water in your mouth, wash, and then take a toothpick or an ice cream spoon or a matchstick. Scrape the inside of your cheek. You have to remove a part of the skin from your inside of your cheek. Now that scraping in a drop of water on a clean slide. Now spread it, and what type of dye we are going to use? That is methylene blue, or we are going to use iodine. So these are the names which you have to study. Then cover that part with cover slip, remove excess of stain with the blotting paper, and then see it under the microscope. So this is the diagram of your cheek cells. So what do you see? A number of scattered cells are seen. As an onion, it was in the form of bricks lying together. Now each cell is polygonal, irregular in shape, and you have in the center darkly stained nucleus and a thin plasma membrane surrounding each cytoplasm, cell cytoplasm. So vacuole and cell wall are absent in the animal cell. You will not find here cell wall or any vacuole as we have seen in the plant cell or the onion cell. Now third activity is to study blood cells. So this is different type of, take a drop of blood, put it on the slide, cover it and then see it under the microscope. Amoeboid shape, that means which can change the shape. We call it as amoeboid because it is the word coming from the word amoeba with clear distinct nucleus. See, these are all different types of nucleus, right? So these are known as WBC or white blood cells, and concave cells without nucleus. So this is mature RBC, concave in shape, it doesn't have nucleus. Mature RBC do not have nucleus. This is one difference between them. Then blood platelets are too small to be seen under a compound microscope. Now this is unicellular organism. Observe them. Amoeba, this is amoeba and this is chlamydomonas. Very important diagrams. Draw the diagrams and then you see the difference between the two. Amoeba is irregular. Finger, see, this is pseudopodias. It can come here, it can be formed here, anywhere you can form them. And cytoplasm is jelly-like, colorless, transparent and contains a single nucleus. Amoeba also has contractile vacuole and many small food vacuoles. A single circular nucleus is present in the center. On the other hand, chlamydomonas body is simple, unicellular, spherical, and cytoplasm is surrounded by plasma membrane and cell wall is also present. A cup-shaped chloroplast with is also present. If we see this cup-shaped, see, a cup-shaped chloroplast is present in this. Two flagella are found for locomotion and an organ called stigma or eye spot is also present. Nucleus is enclosed by the chloroplast. See, nucleus is enclosed by the chloroplast. Two flagellum, eye spot is also present. Okay, so these are the here we finish your chapter and this is the checkpoint. You can do it on your own. Hope it is clear to you.